Good afternoon, students, and we are studying protein synthesis. Uh, if you have got your textbook with you, please open the page number 77. Okay. Last time I told you the first question. See here, you can see on page number 77 on the right side, you can see the title protein synthesis. Here is the title protein synthesis. And the question is below this paragraph. I have written this question. What is the what is the central dogma of protein synthesis? Central dogma means central idea, central theme. So protein synthesis essentially consists of only two steps, and these two steps, they form the central dogma of protein synthesis. And these two steps involved in the protein synthesis are, this is the first step, that is transcription, and the another second step is translation. Inside the nucleus, DNA first produces mRNA molecule, and this process is called as transcription. And this mRNA then comes out of the nucleus, enters into the cytoplasm, and in the cytoplasm, this mRNA produces protein molecule or the polypeptide chain, and this process is called as transcription. So there are only two steps involved in the central dogma of protein synthesis. First step is formation of mRNA from DNA in the process called transcription, and second step is formation of protein or polypeptide from mRNA in the process called as translation. So only this much you have to give in your answer, or there can be a diagram, sketch and label the diagram showing central dogma of protein synthesis, and you have to sketch this diagram. In words only you have to draw this diagram using the words DNA, transcription, mRNA, translation, polypeptide. And if there is a question, what is the central dogma? The answer is unidirectional flow. Unidirectional flow means flow is in this direction. DNA will produce mRNA, mRNA will produce polypeptide. Reverse flow will not be there. The polypeptide protein will not produce back mRNA, and mRNA will not produce back DNA. So this is unidirectional. The unidirectional, in only one direction. Unidirectional flow of information from DNA to RNA and from RNA to protein is called central dogma of protein synthesis, and it was postulated by F.H.C. Crick, or you can say simply F.C. Crick. Okay. Now, though it is mentioned here the flow is unidirectional, there are certain exceptions, and the exception is RNA viruses. There are certain viruses, and these microbes, these viruses are the RNA containing viruses which produce DNA from mRNA. Means it is reverse is the process. The usual direction is DNA to mRNA, mRNA to polypeptide, but this is reverse direction. Means in viruses, certain viruses, mRNA produces DNA, and this is called as reverse transcription. And this is the exceptional thing to note here. And there can be a question on enzymes also, and regarding the enzymes, the enzyme required for formation of mRNA from DNA is called as This is called as RNA polymerase enzyme. RNA polymerase produces RNA from DNA, and the enzyme which is required for replication of DNA produces again DNA, and when DNA produces its own copy, the enzyme required is DNA polymerase. So you should remember all these enzymes. DNA polymerase is required for producing DNA from DNA, means for DNA duplication or for the replication process. Enzyme RNA polymerase is required for formation of the RNA molecules from DNA, and there are certain enzymes which are required for synthesis of protein from mRNA, and several enzymes are required for this process. Not any single significant enzyme is there. Group of many enzymes is required for synthesis of protein. Okay. You should remember one more thing. There are three main types of cellular RNA inside the cell. Three RNA types are produced by DNA, and these are rRNA or ribosomal RNA, mRNA or messenger RNA, and tRNA or transfer RNA. And the enzymes required for producing RNA are the polymerase enzymes, and there are three different types of RNA polymerase enzymes. RNA polymerase 1 is required for producing rRNA from DNA. RNA polymerase 2 is required for mRNA production. And this is important as far as the protein synthesis is concerned, because DNA produces mRNA and mRNA produces protein. The enzyme required for producing mRNA is RNA polymerase 2, and RNA polymerase 3 is also there, which is required for producing the tRNA from DNA. So DNA transcribes rRNA using the enzyme RNA polymerase 1. DNA transcribes formation of RNA from DNA is called transcription. DNA transcribes mRNA using the enzyme RNA polymerase 2, and DNA transcribes tRNA using the enzyme RNA polymerase 3. And regarding the reverse transcription, mRNA produces DNA. mRNA produces DNA, and there can be separate question on this. And if you refer to the next page, that is page number 78, here you can see I have written the question here. Which enzyme? The question is, which enzyme allows reverse synthesis of DNA from RNA? This is called reverse transcription. RNA made by DNA is called transcription, but here DNA is made by RNA, and that is called reverse transcription. And the name of enzyme is, if there is question for 1 mark, you have to mention the name of enzyme. The name of enzyme is enzyme RNA dependent DNA polymerase. RNA dependent DNA polymerase. DNA is produced by DNA polymerase. RNA dependent DNA polymerase produces DNA from RNA. Now moving to the actual process of protein synthesis. As we have seen in the central dogma of protein synthesis consists of two main steps. One is transcription and other is translation. Transcription, producing mRNA from DNA is called transcription, and producing protein from mRNA is called translation. Now let us see what the transcription is. Okay.